Okay, so welcome back to Final DSE, and we're now at chapter 27. So from basic genetics to molecular genetics, and now we advance to the biotechnology. In biotechnology, we will talk about GM. So what is GM? GM is genetically modified, and this is also a GM in the Gundam series. However, we're not talking about that particular GM. We talk about our GM, this is genetically modified. So how can we introduce a genetically modified organism? So we undergo genetic engineering, that is a direct manipulation of DNA states. We change them, and usually we will undergo the recombinant DNA technologies, that is we combine those already exist DNA together. So what is the concept behind? So first of all, we obtain the gene of interest first, that is in a form of DNA, and then we obtain the vectors, usually in a bacterial plasmid, and then we cut and paste them together. So we cut the DNA fragment and also we cut the plasmid with the same restriction enzyme. So in this case, it's the EcoR1. EcoR1 cut every GAATCC they encounter, and then we join the DNA fragment and the plasmid together using the DNA ligase that we call the ligation. And then we can have a recombinant plasmid. So what are the applications of the recombinant DNA technology? So for medicine, we can produce insulin and also gene therapies. So there is a, based on GM modifications, we can produce insulin from bacteria instead of we purify insulin from like cow or pigs. So it costs much less and also is much cleaner with respect to the extraction process. And then in the agriculture and also food production, like the soybean and also maize, they are usually genetic modified in order to make it look prettier and also look bigger. And in the industrial use, we can also use to produce certain enzymes. And in the environmental production, the GM oil-eating bacteria can help up to clean the oil spills. And then for DNA fingerprinting, because between our human there are something unique, so it can be used for identifications. The term unique is a very special term and you have to spell it. And how we can identify individuals is by using the gel electrophoresis. By nature, DNA molecules are negatively charged, so they will move towards the positive end. And for shorter DNA fragments, they will move faster in the gel electrophoresis. So based on the pattern of bands according to their size and with respect to the control, that's the marker, we know the patterns. And the gel and the DNA are all standard. And then afterwards, we take a photo of it and then we can compare. So what are the applications of DNA fingerprinting? It can be used in the forensic science. In this case shown in here is a crime scene investigation, the CSI series. So like 10 years ago, most people enjoying American soap opera, they all watch CSI series. And they can also be used for knowing who is the parents and also identifying victims. And authentication of the food, which means that whether the food are actually produced in their site of production, because different sites of production may have different DNA patterns. Then it can also be used in the conservation of biodiversity. And most importantly, the DNA fingerprinting can use to trace the source of disease. In our most recent case, the COVID-19, the difference between the alpha strain, beta strain, delta strain, and also the Omicron strain, this is all done by using the DNA fingerprinting technology. So based on that, we then developed the Human Genome Project. Its goal is to complete the set of the DNA in humans. It started in 1990s and it is now completed. The goal is to determine the base sequence of the human gene and locating all the genes. Okay, because in the previous two chapters we know that there is a one gene, one protein hypothesis. By knowing the ATG, we can identify the genes and also their possible proteins. And then based on that, we can develop new technologies and also try to solve some social issues. And the benefit is to first of all we understand more our genetics, and then we can use the information to diagnose or to treat disease like cancers, and also have a better understanding of evolution. Like it claims that all the humans are from Africa. So by comparing the genome of the African and also the Asian, we can have some ideas about the evolution patterns. However, there are also some limitations of the HGP program, which means that the functions of many genes are still unknown, even though you know that there is a gene, but the function is unknown. And the data obtained are not enough to understand some biological process, because you just know it there is a gene or there is a protein. So here is an extract from the HKEAA document, and we have gone over the recombinant DNA technology, that is the knowing the gene of interest and also cutting the plasmid and then ligate them together. And then for the DNA fingerprinting, it involved the gel electrophoresis and also staining. And we also talk about the Human Genome Project. And if you are really interested in knowing more about biotechnology, I hope your teachers will talk about an elective part, that is the biotechnology part. So this will be the end of the core part of biotechnology, that is for the chapter 27. I hope to see you guys next time. Bye.